This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 866, recorded on February 11th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon DePommier.
Hello, Vincent, and everybody else. Now, the weather report today is that I know you're going to feel somewhat insulted when I say this, but I wish I was outside instead of here right now, because tomorrow is going to be even better, but the next day is going to be worse. So today, it looks nice, and it is nice. It's in the high 50s. This is a very anomalous event. It's February, 12th? 11th. This shouldn't be happening.
Well, it's going to get cold again. Don't worry.
Yeah, I know that. But
Dixon, I'm going to come to your house tomorrow. We can enjoy the warm weather on your terrace.
We can enjoy it. We can have a good time.
But we do have work to do. I'm going to Dixon's house to sort out his computer.
His multiple issues.
Okay,
maybe we'll solve some of them.
And plus, I'm bringing a print that Michelle Banks made for me, and he's going to put a mat on it, and then it can hang in the incubator. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It is a glorious day. 74 degrees and sunny. It is absolutely glorious.
And from Madison, New Jersey, Brienne Barker.
Hi, everyone. Not quite as glorious as Rich's, but pretty glorious for New Jersey in February. 58, sunny, and just lovely.
Also joining us today from New York, Amy Rosenfeld.
Hello.
Welcome from 138 blocks north of here. I assume the weather's the same, right?
You were here this morning?
I was there this morning to take care of the mice, right?
Yeah, we had to bleed before we make hybridomas.
And much of what we're going to talk about today, that's when I used to go out, when I go, Tuesdays and Fridays. Tuesdays boost, Fridays bleed. By the way, what was I going to say? And I forgot. And I also made sure your computer was ready for today's TWiV, right?
Yes, that's because it was broken.
And we fixed the lighting because some guys said that we could light you better. So I'm lighting. And then I said we should also light Brienne better. So Brienne's getting a new light on Monday.
I know. I'm very excited.
The problem with my light is if I light me better, it makes me squint.
Okay,
so
you want a light too? I can get you a light, Dixon.
You can get me a light.
Yeah, there are these very nice lights that sit right next to you on the table, and they cast a nice glow.
Can you get me an outlet also?
You don't have any. Well, the AC I can't do. Okay, now we have a public service announcement from the annual Biomedical Research Conference for Minority Students. They are launching a new meeting, the ABR-CMS, that's their abbreviation, e-poster spring symposium for emerging scientists for selected high school students who've conducted formalized research, community college students, and first-year undergraduates to present research in one of the 12 ABR-CMS disciplines. This meeting, which is held online Saturday, April 23rd, 2022, will help students prepare and qualify for subsequent ABR-CMS. Hey, look, it's your initials, Amy, ABR. ABR-CMS participation. Oh my gosh, CMS is Chris Schindler's initials.
Amy, what does the B stand for? What does the B stand for?
For me?
Yeah.
It's Amy Beth. Amy
Beth. Aha.
Amy Beth. But it's Chris W. Schindler.
Okay, it's W. Someone is CMS. But you have two biblical names, don't you, Amy?
Yes.
I'm sorry.
It's ABMR, yes.
ABR-CMS participation in the annual fall meeting. Abstracts are due May 15th. You have a little over a month.
March 15th.
Yeah, sorry. Oh, this is terrible. I see one thing and read another. March 15th, 2022. Thank you. In addition, ABR-CMS wants to promote the professional development of early career scientists by encouraging graduate students and postdoctoral scientists to sign up to be abstract reviewers and judges. although all active researchers are eligible to volunteer. ABR-CMS 2022 is scheduled to take place November 9th through 12th in Anaheim, California. Additional details will be available in the spring of 2022. And there are links to this that we will put in the show notes for you.
This looks like an important organization and important events. We need this.
Their ABR CMS meeting each year, the one they mentioned happening in Anaheim, is a great meeting. I think it helps out a lot of students and is a really wonderful thing from what I've heard.
All right, so check it out. And right now we have a paper here today, which is the work of Amy B. Rosenfeld. And so we're going to talk about that because it's quite interesting. And of course, we have the author here to discuss it with us. So this was published not too long ago in MBio. It's called Cross-Reactive Antibody Responses Against Non-Poliovirus Enteroviruses. And Amy's the first author, and then Edmund Shen, Michaela Melendez, Nisha Misra, Ian Lipkin, and Vincent Racaniello, who are all at Columbia University. So, Amy, who are these people first before we get into the topic?
Edmund is our undergraduate. He's graduating this May. So he's been with us theoretically for 5 semesters. He came, or 4 semesters. He came January 2020 and went home January 20. And then he returned around September, and then he went home again, and he came back. Michaela was our tech for a while. Nisha is an assistant professor with Ian at the Center of Infection and Immunity, and then Ian is the head of that. And then there's you. Okay.
So Edmund took my virology course when? The year before? Is that correct?
No, Edmund took your virology course in the spring of 2020.
Oh,
and on day 2, he came to talk to us, but he found me. And he's been with me ever since.
So I should call that Virology Interrupted.
Yes.
There's
Virology
latent phase.
No, there's some play or something, Interrupted. Is it Girl Interrupted?
Girl Interrupted.
It's a book. Yeah, Virology Interrupted. All right, so the thing about this work, Amy, is that you started it really about a year ago, correct?
Yeah, maybe March.
And we started immunizing mice and bleeding them. And so why did you start? You were working on different things, many different things, right?
Yeah, we're always working on many
But this things. One, where'd you get this idea from that there's so there are a lot of different enteroviruses, and you should tell us about that. Where did you get this idea from?
So, there was an RFA. We had gone to a meeting. There was an RFA about looking at cross-reactive immune responses to enteroviruses.
Okay.
And we've known for a while that there are a lot of enteroviruses out there. There's been more than 110 identified. And we know that they have cross-reactivity. So Michael Wilson and Nisha and Ian have both published papers using peptide arrays and passing serum from healthy individuals over them. And these arrays of peptides are composed of peptides that are derived from the capsid proteins of enteroviruses. And you can always find antibodies that bind. And many people don't know that they have any entero infection. They're perfectly healthy. And so we've known about that for years. And also, you and I have continuously had ongoing disagreement about how many serotypes of certain viruses there actually are. And what is a serotype?
And what is a serotype? I want to ask you to define that in your word, because we have arguments because my definition is like 80 years old.
Well, your definition is just wrong.
Okay. What's a serotype?
So it goes all the way back to and I'm really kind of surprised that it's so far off, actually, because it goes back to David Bodian and polio and the observation of secondary attacks of polio infection and paralysis. So he wrote a kind of a seminal paper and did some seminal experiments in the 40s, but it really was actually in 1931 by Burnett and McNamara. But they had known for years that you could get infected with polio, not develop disease, and then at the next outbreak, you would get infected again, and then you would get, and some of those children would develop, or some of those patients would develop paralysis. And this was like, in 1939, they had like 26 secondary attack cases of polio paralytic myelitis.
So are you defining secondary attack as you've had it before, you get it again?
You had it before, you didn't develop paralysis, you recovered, and then you come back several years later and you get reinfected.
And what year are we talking about now?
So it's been observed since 1905 when there was the Vermont outbreak.
But how is polio defined at that period of time?
It's defined by muscle weakness.
But if you get it and you don't get paralysis, then
you
how
have
is
muscle weakness.
Oh, you have muscle weakness.
Right, and then you recover.
And so that's how we're defining infection here, obviously.
Yeah,
well, we're not defining infection. We're defining disease.
Because there was no way to. Well, you could take.
You can't say, because most people were infected get febrile disease. They get a fever. So we're defining it as the protection against what is severe disease.
Was there any way to, in 1910, 1920, could people look for antibodies in your blood? There were no cell cultures, so we couldn't really.
They looked for antibodies, but they did it using animals. This is where Flexner kind of screwed up.
Yeah.
Also.
So when year-wise was polio identified as a virus? When could we say this is a virus that is causing this disease?
1905.
Yeah, 1905.
By
the German.
Viennese guy who discovered the blood groups. What was his name?
Landsteiner,
thank you. Yeah,
is it then defined in an animal model as a filterable agent that causes disease in animals?
Well, no, because it was very hard to passage until they were able to grind up a brain and passage through the monkey. They had to find a host first. It wasn't very clear. It took a while to find that host.
The kid had died of polio. He took the spinal cord. He ground it up. They knew there were lesions in the cord. He ground it up, and he happened to have some old world monkeys in his lab. And he put it into their brain, and they got paralyzed. And
that's 1910?
1905.
1905.
And was that, did he filter it? Did he know it wasn't a bacteria? Okay.
Yes. Yeah, sorry. I left it out. He filtered it. Yeah.
Yeah.
Okay.
So then it got overshined by Flexner, our favorite.
Well, Flexner was at the Rockefeller. Thought he could make a vaccine, but didn't know there were more than one serotype, right?
Well, he didn't know lots of things.
Yes. He knew a lot about medical education.
Yeah, apparently.
But actually, Flexner went to he was poor. So he was like a boxer. He was like a street fighter from Brooklyn or something. And then his brother-in-law got him into medical school, which was basically like a mail-in catalog medical school. That's how medical school was done back then.
Sounds like a good idea.
I mean, yeah, he was if you read the polio book by the guy at NYU, Flexner was not a genius. He was a bully, but he was not a genius.
Oh, Oshinsky, yeah.
Yeah, he was not a genius by any means.
There's a building named after him at Rockefeller.
Well, yeah, because he basically made himself president and then booted out anybody who threatened or disagreed with him.
Was he editor in chief of the Journal of Experimental Medicine?
You mean like Peyton Rouse?
Yeah, like Peyton Rouse, and he rejected anybody's papers who didn't agree with him.
Well,
Anyway, we're diverging.
So this conversation starts out with trying to define what a serotype is. So now I know sort of the context of what we knew about whether or not this was a virus or whatever. And you've told me that apparently you could get a polio type disease twice.
Yeah.
Okay. So how do we get from there to serotypes?
So they had virus, and so they had an animal model that could recapitulate disease, which is critical. And it's absolutely critical. And so they took these viruses and they put them into animals. And then they would come back and they would challenge them with the same virus or different viruses. And this is really work. The seminal paper, as I said, was done in 1931 by Bennett and McNamara in Australia. And they had a very hard time deciding whether or not there was actually more than one serotype, because it's not an absolute thing. But they had 3 viruses that they worked with, and they could define that you could not protect an animal if you gave virus A and then virus B. But if you gave virus A and then came in with C, you could protect it. And then it was repeated by Paul and Track. And then it was flushed out even further in the 50s by the Infantile Paralysis Committee, which is like the March of Dimes.
Yeah, they're the one who actually decided there were 3 and they needed 3 in the vaccine, right?
No, it was decided before the infantile paralysis. So people
they funded Salk. They funded Salk to do that survey, no?
No. It was defined before, because in 1949 they had discrete, so then they made what they thought was the prototype virus. So for like type 1, it wasn't Mahoney, because Mahoney was too virulent. It was Brunhilde. And then
that sounds like an opera.
Yes,
and then 2 was Lansing, and 3 was Surrett. Right? Sauquet.
That's like a place in Wisconsin or Michigan or something.
And are those
Lansing is in Michigan.
Yeah.
Are those still? We're now late 40s. Are those still serotypes defined
they're the prototypes?
And they're still
serotype.
Defined by cross protection in animals.
Yes.
Okay.
So you would take samples from patients. You would infect the monkeys. You would harvest their spinal. You would harvest their antibodies. Then you would go in and infect. Then you would boost. You would immunize the next monkey, and then you would go in and infect with the prototype. So it took 3 monkeys, basically the prototype of what you thought was type 1, the 3 types, and you would see if the monkey developed paralysis and died, or if it was protected. So it's defined by the ability to protect against the development of paralysis.
Okay.
So Richard, severe disease.
For pox viruses, do you use the idea of serotype at all?
Yes. But I'm, because, oh man, I would have to dig back and look at that. Because, I mean, we don't talk about serotypes much, put it that way, okay? Most of the pox viruses are different enough from each other so that it doesn't really come up. Like there's not multiple serotypes of variola, for example, okay? Or, as to my knowledge, multiple serotypes of various other sort of avian pox viruses or something like that. So it's not something that is, though it gets talked about from the point of view that, let me see, yes, most of what we call orthopox viruses are cross-reactive, okay? So serum from one can neutralize another, and that, in fact, is, I think, a major criterion for grouping those together as orthopox viruses, even though in other ways they have quite different characteristics.
Okay. Now, Amy,
but serotype is not absolute.
Yeah. Well, yeah. That's other viruses have since been serotyped. Like there's one serotype of measles and so forth. But then at some point we stopped using serotype, right? When the genome revolution kicked in, we just call it genotype now, or nobody talks about serotypes of SARS-CoV-2, for example.
No, you talk about genotypes.
So
You have to define what is going to be the criteria of a serotype.
Yeah,
is it going to be the criteria of protection? It traditionally is the criteria of protection against the development of severe disease. So if you don't have an animal model that can recapitulate severe disease, you cannot define serotype.
Yeah, because I've always naively, I guess, thought of it as defined by reaction with antibodies. Okay. Cross-reactivity. But I mean, that's where we're going with this, right?
So that's why.
But in actuality, that's not really what it is.
Right.
Yeah,
because they just took serum from patients who recovered or whose viruses, who had polio. They immunized an animal and then they challenged it with another virus. They didn't purify out the antibodies.
Right.
And who knows what's in that gamish that they put into the animal, right? You have T cells and all this other stuff. So we have narrowed it down and have used the word most likely, probably incorrectly for many years, defined and put it only as antibodies and have made it an absolute thing. So much so that like we've had discussions or we've gotten comments about like, there's minimal protection from polio 1 to polio 2. And it's actually because of that comment that I went back and I read all of Bowden's, Paul's, and McNamara and Burnett and all of those papers that were published between 31 and 55. And there's close to 250 of them where it's not absolute. There are isolates of polio that can not protect against others, but can protect against, a different combination. And also Chumakov's antibody was actually really a propellant for that.
So
I think this is really useful, Amy, because I'm now realizing that there is almost like a piece about immunodominance and things like that that's a part of this definition that I never appreciated before.
Immunodominance is different because you talk about an immunodominant epitope.
Right, but I'm saying that in some ways if something is able to protect against severe disease, you're probably looking at transferring an antibody that binds a dominant epitope.
It doesn't need to be an antibody. That's my point.
Right. Whereas otherwise you might be transferring other antibodies that are not protective, maybe because they're not dominant. And so I hadn't appreciated sort of that level of specificity. And so I find this very helpful.
Yeah. My real point is so you kind of fell into the problem is that you've defined it as an antibody. It happens that it is probably an antibody, but without correlatives of protection for polio and stuff, you can't really say that. But when you just take a serum from a patient and you put it into an animal and then you challenge the animal, it could have been a T cell.
But don't they centrifuge out the cells when they do that?
Well, he doesn't really specify. They just ground it up.
Okay.
They talk about, we had a Waring blender of, with this 4, VPM motor. And I'm like, a what? I mean, I have a blender, but I don't know what its motor is. Like, who knew these things?
By the way, that was named after Fred Waring in the Pennsylvanians, but let's not diverge. How different genomically are the 3 different, what shall I call them?
Types.
Of polio, you've got 3 different polio virus things.
Types.
How different are they?
What is it,
genomically,
like 3%?
if you compare their genomes.
I think it's, 3% if you look at the genome?
That's
it's pretty similar.
Yeah.
And yet they're anagenically distinct.
Yes.
Now, Amy.
So,
Oh,
I'm
sorry.
sorry. I just
but
need to ask one more question.
Yeah,
If
sorry.
you're exposed to all three at once, which one dominates?
Type 2.
Type 2.
From
And that's a capsid difference?
So actually, I was asked that question a few weeks ago, and I kind of flubbed it because I was like very nervous, and
Here's
I kind
your
of
big
flubbed
chance.
it. Yeah, here's my big chance to redeem myself to like a few of the questions that I flubbed it like 2 weeks ago when I was a bit nervous. But yeah, type 2 dominates, which is why
because
I think it
tell us
replicates
why
a
it
bit better.
dominates. Tell us why
Doesn't
it
it
dominates.
have better replication kinetics?
Yeah, that's why you have to get 3.
So, yeah, that's what I was going to say. That's why you get
one dominates,
3. You as a kid you get 3, 2, 4, and 6, and then you get 1, 2. You get 1, 2, and 5 for being fully. 1, 2, and 5 for being fully protected.
Amy, so the rhinovirus, so enteroviruses include rhinoviruses, right?
Now they do.
Counterintuitively, but
well, they do based on VP1 classification, right?
The genome. I mean, they used to be in the old days. We classified viruses not on the genome sequence because we didn't have it, and they said, "Well, rhinoviruses infect the respiratory tract, and poliovirus is the enteric, so let's call them rhinoviruses and enteroviruses." And then the genomicists came along and said, "Nope, they're all the same."
Well, when I was a graduate student, you had REACH for rhino, entero, aptho, cardio, and hepato.
That's right. Only those were the only genera. Anyway, my question is, so they used to call them rhinovirus serotypes, and then they stopped doing that. And so when you get a new rhinovirus, you just make it a genotype based on the genome sequence, right?
No, just based off of VP1.
VP1 only, the capsid protein. Okay. So before we go on, can you tell us what other
serotype of rhinovirus? They did them by challenge experiments in volunteers a lot of times.
What kinds of enteroviruses besides poliovirus and rhinoviruses are there? Just so that people know what you're going to be talking about.
So Coxsackies. So there's Coxsackie viruses A, B. Mm-hmm. There's echoviruses, which are enteroviruses, which lack cytopathic effects in humans, and so they're orphan viruses. They're enterobes.
Well, they were originally thought to be orphans, but they're not, right?
Yeah, but Sabin, when he identified them, called them enteroviruses lacking cytopathic human orphan viruses.
And they were enteric cytopathic human orphan viruses, ECHO.
Yeah. Yeah.
Just rolls off your tongue.
But they're not orphan.
They have disease associated with them.
Shouldn't have used that word anyway. It's just unfortunate.
Orphan used to mean no disease association.
Yeah.
Right.
So, and as a group, the enteroviruses, as a group, the enteroviruses comprise a spectrum of disease phenotypes. Is that correct?
Yes.
So
they're very broad. Secondary sites of infection are very broad and result in very different things.
So are they all initially respiratory?
No. Most of them are gut.
Most of them are gut. And
which is confusing. That's why they're called entero. So they were initially gut viruses. And then when we identified like 68 and it was called entero D68, then became rhino 87. Then under genotyping, it became back to enterovirus 68. And because it has entero in the title of it, people think it replicates or infects the gut. It's kind of unfortunate that we initially identified viruses and gave them names based off of where we thought that they replicated.
So, okay. So it's the naming that's the problem, really.
Yes.
Okay.
Yes.
But we have things that are called enteroviruses where the pathology is confined to the respiratory system, correct?
Well,
and we
no, because
rhinoviruses, yes.
Well, rhinos are confined to the respiratory system.
And yet other enteroviruses that are truly gut viruses, and the replication is confined to the gut. Is that correct?
Not fully confined to the gut, because like ECHO-1 can replicate and Coxsackie can replicate in the heart.
Okay.
And then there's enterovirus D70 that replicates in the eye conjunctiva, causes a hemorrhagic fever. Coxsackie also can replicate in the eye conjunctiva. And then lots of Coxsackies cause aseptic meningitis. And there's a thought that Coxsackies cause, or if you're a child, that they can replicate in beta cells of the pancreas and lead to type 1 diabetes.
So a given virus can. I'm assuming that some of these are quite specific in terms of disease phenotype, and others are more generalist, can cause several different disease phenotypes. Is that correct? In other words, do you have some that are, say, strictly respiratory viruses, and some that are both respiratory and gut and conjunctiva and et cetera?
Yeah, I guess. I mean, rhinos are really restricted to the respiratory tract, right?
Okay.
But like Entero 71, it replicates in the gut. It really initially was associated with skin blisters. So it's referred to as foot and mouth disease, right? In children. But now we know it can also lead to paralysis, so childhood paralysis. So it can go into the CNS and cause a polio-like disease.
Okay, good.
You think we should rename this genus because it's so confusing to outsiders?
Well, Vincent, allow me to interject some biology into this, because when you start struggling with the concept of species and genuses, it requires more biology than the virus actually possesses. Viruses are monkey wrenches, remember, right? You were the one that taught me that. So what you're dealing with is a series of related monkey wrenches to various cell types in various parts of the body that you're trying to force into a system that was built around sex. And sex drives species.
Okay.
How would a biologist actually talk about the differentiation of, let's say, SARS-CoV-2 that has how many different mutations in all of its parts? When you add those all together, how far away from the original do you have to go before you're going to declare that another species?
Yeah, so
you're not going to go there. You're not going there.
This is one of the things that makes the virus taxonomy confusing.
I'm not confused.
Is that virus taxonomy uses words that are used in Linnaean in biology, but the words don't mean the same in virology.
No, they don't. But now you're stuck with serotype and species.
Right.
I believe our audience is becoming really confused at this point.
They argued for years over the definition of species in virology, and I think the definition they came up with doesn't really apply anymore now that we are in the genomics era.
Yeah, I think that's actually in a lot of parts of microbiology, not just virology.
I'm
not sure that some of the bacteriology parts of
take heart in the fact that every year that the Linnaean Society meets, they still argue over what a species is. So you can't make anybody happy with this conversation.
Yeah. Okay.
Hey, so Amy, why don't you just walk us through the experiments? A big part of this paper is plaque assays, right?
Oh my gosh.
So there's 2 experiments, right? It's either an ELISA or a plaque assay.
There you go.
There isn't anything in between.
Inevitably, when I walk into the lab, no matter what day it is, Amy is either counting plaques or staining plates or doing a plaque assay.
Outstanding.
Yeah.
Yes.
So now that we're back to the paper, Amy, give me once again the motivation for this. What are we trying to find out?
So really, I wanted to know whether or not there was cross-reactivity, and what serosurveys actually mean, and if serosurvey data is useful in understanding what virus is you've been exposed to. Can I actually say that you have been infected by X definitively or not?
By just looking at your antibodies.
Right. because
without looking at the genome.
No, there's no virus. I don't know when you got infected. And the problem with enteros, for the most part, is that they're very mild. Like they cause a fever. Yeah, most of them cause a fever or something that's very mild. You don't even realize you're infected, or like the cold causes a common, mild congestion, and you go on your day and you never go to the doctor or anything. It's when you get when it disseminates to the secondary site of infection and you get the severe disease, do you go?
So the dogma was, Amy, and correct me, I'm sure you will correct me if I'm wrong.
I love you too, sweetie.
The dogma was, if you had antibodies to polio, they would not neutralize any other enterovirus. Is that correct?
Right.
Okay. And you showed that to be wrong.
It is an indication that you had a previous polio infection. And the problem with that occurred in 2014 with the initial outbreak of EVD68. Or resurgence of EVD68.
Yeah, it seems to me that from reading this, one of the big deals is that people dug up serum and saw that it reacted with EVD68 and said, "Oh, you had an EVD68 infection."
Yes.
Okay, and the question is that true?
Well, the question is, if you did, there's no indication that it was circulating.
Right.
At that point in time.
Right. So did you?
Right, exactly. Okay. Did you really?
Great. Cool.
So it's an extension of it's an extension or more comprehensive reductionist way of answering this question, because it's very hard to do if I don't know what you've had previously, right? But now I've developed a controlled system where I know what virus my mouse has had, and now I can ask, is there cross-reactive antibodies, and are those cross-reactive antibodies neutralizing? Because binding also you cannot extrapolate neutralization from binding, which is another problem.
So what did you actually do? You collected enteroviruses?
Yeah, we've collected enteroviruses for years. Every time that there's a new available isolate of 68, I like to have it because there's always something. Because part of 68's hysteria is that it evolved a neurotropic phenotype, which we had shown in our first publication that was not true. But I always like to have the latest and greatest. You have to have accessories.
So how many enteroviruses you got, Amy?
Today in the paper, or just in general in my
your
collection?
Freezer?
In my freezer?
Yeah.
I have 111. But they're not all individual. Like I have 32 isolates of EVD68 spanning from the original outbreak of 1962, some clinical isolates that a collaborator sent us, and then almost every isolate that BEI has.
And so of those 111, how many are you using in this paper?
I think we used 18. Because some of them I acquired after we started, and there's just so many mice that I can keep track of also. And the mouse cage is actually the most expensive piece of property in New York City.
What's
At it's $1.17 a day or something.
For 5 mice, right?
For 5 mice in the little cage that then we have to come and clean. Yeah. And when you put it in perspective, like I think the most expensive apartment was sold in New York, like at the end of the pandemic or at the end of last year for like, what was it, like 125 or 200 million or something or other.
Put a lot of mice in there.
Yeah.
Their habitats are measured in square inches, not square feet.
So you immunize mice with different enteros.
Each cage is segregated, and that cage is dedicated to an entero, right?
One entero, right?
One entero. So we have monospecific polyclonal sera.
And so on Tuesdays, I would go in, and she'd say, "Okay, we're boosting Coxsackie B, Entero D7, we would do all those. And then on Friday, we would bleed some others, right?
Yes.
And so then you take the serum and you do, and I would bleed and I say, is this enough? And she would say, no, get some more. Or, yes, that's fine. I got to be good at boosting and bleeding.
Everybody has to have a role.
I have to say, there's nothing like practice.
There is nothing like practice.
So bleeding a mouse, in fact, I learned this a year ago from one of the mouse husbandry people. I said, how do I bleed a mouse? Tail vein? She said, no. No.
Now you're going to gross out the listeners, but go ahead.
No tail vein. It clots. There's a vein underneath the jaw.
The mandibular.
Submandibular vein. And you just take a little lance and you poke it and you get a few drops of blood. She said, don't take more than five drops. Because a mouse only probably has 10 drops.
That's right.
And you get it, and it's very good, and then it stops bleeding. It's really very easy. And then you take it back to the lab, you do plaque assays, right?
Well,
how do you do a neutralization assay, Amy?
So we made twofold serial dilution. So we spin out the cells and heat and activate and stuff. And then we take the supernane, is the sera or plasma, if you would like. And then we do twofold serial dilutions, and we incubate with a 10 to the 3 PFU virus. We end over and incubate for an hour, and then we do a plaque assay.
And so you are unlike those previous researchers you mentioned in that we know that you're just looking at antibodies. You aren't using the weirdo blender.
No, I'm not using the weirdo blender.
You definitely have just antibodies.
Yes, I'm not using the weirdo blender because the weirdo blender just freaks me out.
Amy, so when I collect the blood in little tubes, you have EDTA. So what's that for?
So it doesn't clot. I made my own non-clotting tube for
Why
my little
does it
mouse.
matter if it clotted?
Then you'd have plasma, not serum. That's right.
Yeah.
Well, so what's the problem? Would it trap antibodies or what? What's the problem with that?
Well, it takes up the entire volume of the tube. There's no liquid. Because a few times you had to use the wrong tube where it clotted, and then we come back to spin it, and there's nothing to spin.
All right, then
EDTA is a chelator of calcium, as I recall. Is that right?
Yeah.
Chelator, calcium, and magnesium.
That's right.
So
it's not a heparin or any of the other.
But no.
So, Amy, what would you do? You would take, let's say you immunize a mice with poliovirus, right? Mm-hmm. You would take its serum. You would check it against polio to make sure it worked. You have antibodies, right?
Yeah.
And then you check all of your other viruses.
Well, I check as many as you can use.
Yeah.
It's not going to, it doesn't cover all of them. So some of them, we have different ones that were able to be checked. And sometimes you got more serum than others. And when you did an ELISA, you need serum and stuff. You need less because of background levels of against the plate. But sometimes we couldn't do all of them at the same time.
Yeah. Now, as you said before, you do ELISAs too, for just to measure binding, right?
Yeah.
So in some cases, you see binding in the serum to a particular virus, but not neutralization, right?
Right. So there's a lot of anti-polio antibodies derived from Mahoney bind efficiently to Rhino 14 and 39, but they do not neutralize them by plaque assay.
But some polio antibodies do neutralize some rhinos, right?
Yeah.
That blows me away totally. It totally is contrary to how I've been brought up.
Yeah, I know.
The converse, am I correct that the converse is true, that neutralizing antibodies, they must have to bind. They must be positive in a binding assay.
Well, for our work, they have to.
Yeah.
But that does not necessarily, but there are probably neutralizing antibodies that target other cellular, they might target the receptor or something else.
Okay. Sure. Right.
But we're not able to assay them in the lab.
Right.
You need an intact animal. And some of the ones that we said, some of the ones like what I just said, polio does not neutralize 14. If I were to take I'd have to do it the converse way because there's no animal model for 14. If I were to immunize a PVR transgenic with 14 and then challenge it with polio and find out that those mice didn't develop paralysis, then it is not a traditional mechanism of neutralization. So like FC mediated or complement mediated, like you see for Symbis and for other philo and for philos, I'm not assaying for. I mean, this is really basically saying you must somehow be blocking cell attachment and entry.
Right.
So
or
Vincent,
maybe you could say it causes an aggregation of the virus in the solution, and then it's not in the right orientation.
So Vincent, you said that this was sort of blew your mind, that it was different than what you had learned before. Is that the idea of the cross-reactivity that was novel to you?
Yeah, well, I just thought that was the definition, because some of the older people in the field had taught me that. So antibody against polio would never react with rhino. But in fact, I don't think anyone ever checked, right, Amy?
Right.
Right. There's no record of them checking. And actually, there's record. So people tried to develop a diagnostic based on antibodies. And that's kind of like a separate project that came out of this. But so when I did certain experiments, and the pattern fell, like our ELISA pattern, and then I wrote this grant about doing something else related to antibody binding. My pattern was exactly what had been reported in the 90s. And I hadn't even known about the paper. Like the paper was because I read the paper because I was writing this grant on something else. I was like, oh, Vincent, look, this is exactly the same pattern that we just saw. And he was like, really? And when they did those experiments, they didn't say anything about neutralization. They just looked at binding.
I have to say that I was recalcitrant, right? Because,
yes.
And that's the source of many arguments.
You should use EDTA for that, Vincent.
Very good. Very good. And the name would say, no, you're wrong. I said, well, this is how I was trained by Roland Ruckert and other people who said, no, the antibodies, that's a polio virus. It's serologically defined. And Amy she stuck with it, but then she got the data that shows it. Now there's no going around.
Oh, wait, I like that it was the source of many arguments. It was the source of, you have no respect for me. I'm your elder. I've been doing this for 40 years. You are a whippersnapper.
Look, Amy, as long as you didn't start throwing broken glassware at each other, it was a civil argument.
Some people do that, I understand.
I know.
So this is an interesting commentary on science for our listeners, because stuff gets fixed in the lore. Okay, that may or may not have experiment to back it up, and it's very difficult to dislodge it. And one of the things that comes to mind is, in my active life as a scientist, one of the things I did was review papers. Okay, and papers would come along that were just dead wrong. Okay, and it was so important to make sure that they didn't get published, because some of this wrong stuff, if it gets published, gets fixed in the literature, and it takes years to undo it. There was one particular incident where a particular laboratory had an idea that they thought they had evidence for that screwed up the field for 15 years. There's another one that's still ongoing, and it's a problem.
We talk about this all the time.
Yeah.
This stuff gets published, and Amy says it's screwing up the field.
Yeah.
Yep.
But science is a self-correcting.
It is.
It's self-correcting unless you're us and you're in financial distress all of the time, and your reviews for your grants reiterate the things that are just not correct.
There is a human element to this. That's a delaying tactic, but it doesn't stop the truth from eventually emerging.
It's self-correcting. We're just not always
I have no
having it
patience.
self-correct on the time scale.
Yeah.
We might like.
Yeah.
So
It may take
Amy,
generations.
Amy, can you
Yeah, but
have
I
a
don't have
serum?
any patience.
I need to ask a question about the experiments now. You have a serum that has both binding antibodies and neutralizing antibodies, and they're not the same.
I don't know
Can
if
you-
they're the same or not.
No, but you could find out though, couldn't you?
Well, that's what we've. So after we did this,
So wait a minute. I have a question. So can't you use virus to absorb all of the binding antibodies out of your serum or plasma, and then if you've got neutralizing antibodies left that still have to bind to the
They have to
organism,
bind. Yeah.
so you're going to remove everything when you bind everything, right?
no.
So
So
how would you
we were
distinguish
actually
between?
asked to do that. Okay,
How
so
would you-
in the review,
I'm
of the
sure.
paper.
By the way, I wrote that review.
Yeah,
No,
you
that's
might
not true.
have. Maybe you
I
talked
didn't.
it.
No, I didn't.
In
I
the
didn't.
review of the paper, the guy asked us
Yeah,
to
absorb out, right?
absorb all of the antibodies that
And what's
bound
left?
that bound polio, and then take that absorbed serum and try and neutralize like EVD68.
You
And
can't.
the problem with that is my readout is a plaque assay. Okay.
Right.
And polio will grow in HeLAs and RDs, and EVD68 will grow in HeLAs and RDs. So how am I going to, if I get plaques, how am I going to distinguish that those plaques were from polio or from EVD68?
Green fluorescent protein.
Well,
Label one of them with that.
You could do a focus forming assay, right?
we could,
Oh, there you
but
go.
I don't really have an EV. There were problems with doing that. Okay, so like the antibody for EVD68 is anti-mouse. My serum is anti-mouse. There's not really a good way to do this.
So I'm guessing this is why you're making hybridomas.
Right.
So I'm making hybridomas because it's the traditional way, and the actual experiment that I wanted to do is $50,000 for one mouse. And you want to give me the $50,000?
So Amy,
No, I don't have that right on hand right now.
why do you? I think you should tell them what this experiment is. It's very interesting. But why do you need monoclonals or monospecific antibodies?
Because I want to. So you have, in theory, 4 potential options, right? You have an antibody that binds polio and binds EVD68 and does nothing. You have an antibody that binds polio, neutralizes polio, binds 68 and does nothing. You have an antibody that binds polio, doesn't neutralize, but binds and neutralizes EVD68. Right. And now I want to know if there are, so the dogma of the field is, oh, you might have cross-reactivity in a polyclonal sera, but you'll never have cross-reactivity in a monoclonal sera. Because a polyclonal sera is 100, if not thousands, of monoclonal antibodies. So if you have this phenotype in a polyclonal sera, you have to have at least one antibody that has some component of that phenotype.
You want to identify those monoclonals that cross-react.
Right.
So let me see if I understand this. You have sera that cross-react, but you can't tell whether that means that you have.
I can't distinguish the 4 possibilities that I just told you.
But you don't know whether you have, say, 2 different antibodies. One that reacts predominantly with one of your strains and one that reacts predominantly with the other strain. You need the monoclonals to identify whether you have individual antibodies that have whatever of the 4 components you're talking about. Is that a fair statement? Yeah.
It is. But so I'm not very patient. And so I took advantage of other resources to answer that question.
Okay.
So Amy, of your viruses, how many epitopes are we talking about here?
Well, that's another question.
No, it's the question, because when you start monoclonal antibody production, you're going to get it against all of them. How do you know which ones are the right ones?
Dixon. So traditionally on enteros, there are 4 neutralizing antigenic sites that have been defined. And they've basically been defined by monoclonals and using synthetic peptides. Okay. And they fall in, and they fall at the north rim and the south rim and the canyon at the threefold axis of symmetry. And then lo and behold, another conflict between Vincent and myself is the 4th one that falls inside the canyon. No. Rossman said that there can never be an antibody that sits inside the canyon. Do you remember that?
Yeah, but he's dead. Don't diss him.
I'm not dissing him. I'm dissing you.
Which Waxman was that?
I'm still alive.
Yeah,
I didn't think that. I mean, he had done the structure, and he says the antibody, the binding cannot fit into the canyon.
The first thing that comes to my mind is these HIV antibodies that have these long processes that wiggle into all sorts of crevices, right?
Yeah. So it
So
does.
it
Nothing
turns
is
out-
impossible in biology. Nothing.
Nothing. Well, it turns out that several years ago, Konstantin Chumakov at the FDA had identified 4 monoclonal antibodies that are able to bind all 3 serotypes, but will neutralize multiple serotypes, like 1 and 2, 2 and 3, 1 and 3. But you can never find an antibody that will neutralize all 3 serotypes. And then he did the structure of one of these antibodies, which is A12, and he saw it sits in the canyon. And then I recently had a conversation with him, and he has 70 of these.
Wow.
They're not all characterized, but he believes that the majority of them sit in the canyon.
Is he a sharing person?
He's been very lovely. Yes, he's very generous. He sends any antibody that I've ever requested, and he's wonderful.
Is it possible for a monoclonal antibody against poliovirus to neutralize some other virus in your collection?
Yes.
This also blows me away.
We've done that experiment.
A monoclonal. All right, I can see a polyclonal here, right? It's a mixture of all different. A monoclonal, an antibody against one epitope of poliovirus will neutralize. And if you look at the 4 antigenic sites, Amy, on the capsid, how much amino acid similarity is there?
Oh, and this antigenic site, this has no similarity.
None.
But maybe it has a charge similarity. Maybe the charge is the same.
Maybe.
Could be. It could also be structure, but it doesn't. But the assumption right there could also be wrong. It says that because, let's say, it's the antibody that binds the threefold axis of symmetry on polio, you're automatically assuming that it will bind the heterologous entero at its threefold axis of symmetry.
Yeah,
it does not have to bind its threefold axis of symmetry. It could bind at the twofold axis of symmetry. But
it would be a different amino acid sequence.
Completely different locations.
Yeah.
And this was another argument that we had about, like, when I wrote the last grant that went in on Monday. So Eckard and Nemoto, and Phil Miner, who had done a lot of this work for polio and Sabin strains of polio. And actually, Vincent had Phil Miner write a chapter in the book that he edited in 1990. He comes out and he tells you that resistance mostly does not occur at the site that the antibody binds.
This is resistance to the monoclonal.
The
right?
resistance to the monoclonal does not occur at the site that the antibody binds.
I'm not sure I understand what that means.
So it means if I bind on your left hand,
yes,
and you want to select a variant that no longer binds,
do my right hand.
The change does not have to be on your left hand. It could be on your left knee.
I
the way I visualize that is that you could have a change somewhere else that changes the conformation of the initial epitope, right?
That's right.
And it can be distal. And we have looked at that also. And that also blew your mind.
It almost sounds like quantum mechanics.
So if I change my left knee, then that sort of downstream alters my left
All right,
hand.
look.
That makes perfect sense.
Given the repetitive nature of the capsid, distal is not ever very far away, right? Because the subunit is the pentamer, right? And so there are 12 pentamers. So whatever distal changes within that pentamer. So
I don't
it's
know
not like
if from the point of view of an amino acid, distance is, different. Could be quite far away.
Exactly.
I know, but it's not like
it could be a total different neighborhood.
What I'm trying to say
different state.
is this five-fold axis. It's not going to be at the other one on the other side. It's going to be at one of the other five folds around it, or a three or two-fold. That's all I'm saying.
So I have another question related to this. If you could separate, and I know that Brianne has another word for this, and I'm blocking on it. When you separate the two binding sites for a normal antibody, and now you have a single, like a camel, for instance, antibody, does that
Fab.
still work? Yeah, fab. That's exactly what I was looking for. Is it still fabulous, or is it not still fabulous?
Good
Do you
question.
still get the same effect, or do you need both in order to get this difference that you're talking about?
I don't know.
Well, you could find out right away by just simply cutting it in half. You can easily do that, right?
You want me to digest the antibody?
Yeah.
Well, once you have monoclonals, you can
Once I have
enough.
monoclonals, sure.
Yeah, but
But the monoclonals that
are
I
still
have
like
are
this.
But the monoclonals I have,
need one like
Dixon,
this.
I understand. But the monoclonals I have are precious. Some of them, like the hybridomas, have been lost.
Understood.
They're not
got it.
commercially available. I'm not going to. One of the reasons why I made my own hybridomas is because I need a lot of them. I need a large volume of any one that I find is interesting. And if it happens to be an antibody that somebody else supplied to me, I can't go and say, can you give me 100 mils of it?
No, just give me the cells.
Well, that's what I'm saying.
No, they don't exist.
But they don't exist anymore.
I see.
So
Brianne,
somebody is sending, so Costia is sending us the famous C3 antibody that was identified by Gerard, in 1980?
Yeah.
Vincent?
Yeah.
And he says to me, I only have so much ascites. The hybridoma was lost many years ago. I'm thinking about reconstituting it using CHO cells. And I said to him, let me know how I can help.
Yeah, no, I got it. You can just hear Cesar Milstein rolling over in his grave right now listening to this conversation, who I met at a meeting once. He's really an interesting guy, but yeah, these are precious elements. I agree.
Go ahead, Brianne. Go ahead.
So I really appreciate the point that you're making, Amy, about how just because you bind the epitope is on the five-fold axis of one virus, it doesn't have to be on that exact same spot in another virus. I think that that's a really important thing to recall, and I can think of some T-cell papers that show very similar things.
Sure.
As I'm looking at sort of your data and thinking about this model, I have sort of an idea of a question. I don't think it's an experiment that could be done, but I can think of a related one, and I don't know about it. So what I realized from this and thinking about cross-reactivity is that my mind goes to, well, does this mean that your infection history with one enterovirus impacts pathogenesis with another? But then I would say, well, Amy's data shows me that I can't figure that out very easily because I can't tell what past viruses someone has been infected with to really clearly tell their infection history, because I'm going to see this cross-reactivity that she shows. And I wonder, in your mouse models that you're working on, do you tend to see disease when you infect with this? And could you address that kind of question by doing sequential infections in the mice?
So I've thought about that a lot. And I've written many grants to develop an animal model that recapitulates disease and whether or not it can be infected somewhat like the natural route of infection. So like recently there was a paper, a group developed a model of EV71 that can be orally infected, which is the natural route of infection. But most animal models of enteros use some kind of immune deficiency. So they are either neonatal mice that are not really mature, or they are interferon receptor knockout mice, or they're AG129, which is interferon alpha, beta, and gamma receptor knockouts.
Right.
So it's a problem to.
But you could do it with polio transgenics. You could immunize them with, say, Rhino 1A, which cross-reacts.
Right. So that's what we're doing. But it's taken us a while because we didn't have any polio transgenics. You have a blog post telling everybody how you are sacking the last polio transgenic mouse. And I'm going, I don't remember being consulted about this.
That was 10 years ago.
Yeah, what's your point?
Anyway, fortunately, Jackson had them frozen, so...
Yeah, so I'm reconstituting the colony. So this is a problem. So serosurveys are a problem about whether or not you really have the virus that you think that you have. And initially, the work was done by a group at the CDC, and they only asked if you had an antibodies to enterovirus D68. There's no other
Was that neutralizing or ELISA?
Yeah, it's micro-neutralizing.
Okay.
But that's only, their publication only addresses 68. They don't look at a panel.
So you would argue that those anti-68 could have been induced by any of a number of other enteros, right?
Yeah,
so how do you sort it out, Amy? What do you do?
So that's why we took a reductionist fashion, right? That's why we took this reductionist approach where we know that that antibodies that are elicited by immunizing mice are neutralizing in basically the same epitopes as what you identified in humans. That's that's the classic work that Eckart and Nemoto all did. They took peptides from the capsid proteins, and then they immunized their mice, and then they harvest serum, and they did plaque assays.
So I'm sorry, they immunized mice with virus or peptide? Oh, they immunize with the peptide, then they look for neutralizing antibody.
Yeah.
Right. Okay.
And that it's not just for polio. They did this for FMDV. They did this for flu. They did this way before the structures of any of these viruses were resolved or anything.
How big peptide are you talking about?
15 MERS.
And how many permutations of 15 MERS are there per virus?
No, so they knew what the coding sequence of the capsid proteins were. They had the genome sequence of polio. And then they synthesized 15 MERS in certain regions.
How did they decide to do that, though?
Yeah, which ones did they pick before the structure?
I think that they picked by digest. What was the digestion map? It was the science paper, the one I'm talking about.
So they look for things that were on the surface. If you treated the particle with trypsin, they would release.
I see.
But nowadays, Amy, tell Dixon how many tiled 15 MERS you need to cover the capsid protein.
Yeah, I was going to ask that anyway.
So the polio capsid, the P1 region is approximately 881 amino acids.
Forget about it. That's a lot.
Well, I can mass produce it, and I can use, there's a technique that will allow me to do it. I can pull out when I make my hybridomas, I get 250 million viable splenocytes. It's a lot of cells.
That's a lot of wells on a plate, too.
Yes, but if I mix them with a peptide that is fluorescently labeled and with a barcode, I can pull out the B cell receptor. I can pull out the B cells, having that B cell receptor bind to the peptide.
So
I can, what?
How many peptides are you talking about?
So about 850.
So you tile them, right?
So you tile them. You tile them one amino acid, but you have to actually use a bit more because of the way that the capsid works, where the C termini are on the outside and they make C termini of VP1 is close to the C termini of VP2. So it makes a continuous surface. So you have to tile it a little bit. You have to make a peptide that spans that bridge.
So you can have a company make these 850 peptides for you, right?
Yeah.
Got it.
How much does that cost?
I think they sent me a quote of like $8,000 or something.
It's not bad.
The whole experiment comes to about $50,000. $25,000 goes to 10x Genomics, and then the rest is for generating the peptides, the kit to link it to the DNA, and then the sequencing.
So in your lab, you have this little box on the table.
Yeah, my $200,000 box that Ian lent us.
Yeah,
Yes.
Ian bought it and lent it to you. And so tell us what you do with that box. What the hell is in it?
It's a microfluidics device to do single-cell B-cell sequencing based off of binding to this peptide, binding the BCR to the peptide and pulling it out by pulling it out.
Okay, so then the box will give you the sequence?
The box just puts the single cell in a droplet and allows for the RT reaction. And then I have to amplify that and then go to Ian's and put it on the sequencer.
So, Amy, you coat a well with the peptide and then throw in a bunch of cells and then only one sticks? How do you get your cell to stick?
Yeah, the B cell receptor recognizes the peptide.
Right, no, I got it.
It's the antigen. It's the antigen, and then you pull it out with a dynabead. That's at the bottom of the complex.
I see.
So
are we talking about doing this with a mix of peptides or one peptide at a time?
Oh.
Because you
do it with a mix of peptides because at the end of the day, the peptide has a DNA barcode, which is the primer to initiate the RT. And you now know what the barcode sequence is, and you know what the barcode sequence, what the peptide was that correlates to that barcode sequence. So now you have the antibody that is specific to that peptide. And if the structure is resolved, you can go map that peptide and where it is on the surface of the virion. So is it inside the canyon? Is it at the twofold axis of symmetry? Is it one of the loops? What is it?
Basically, and then you have the gene for the antibody, so you can make the
whole antibody gene because I'm not just sequencing the variable region, I'm sequencing through the heavy and light chains.
And nowadays, if you've got the whole antibody gene, you can create a cell that will express that antibody. You can make more of that antibody, right?
Yeah.
Yes.
Yeah. And so now I'm able to, we started off with I would assume that everything we've looked at is IgG, some form of IgG. But for respiratory viruses, it's IgA. That's really important. Mm-hmm. So you do this, you immunize your mice, and I can pull out the IgAs that I'm interested in for a different question.
Brianne, that's why months ago I asked you the kinetics of
you did
G and I, and in fact, when I was looking for your address, you told me, yeah.
But going back to earlier in our conversation, if you identify a B cell from this protocol, so you immunize a mouse, you get the B cells, you react them with the peptides, you now have a B cell that reacts with a given peptide. That doesn't necessarily tell you what the epitope was.
Yeah, because the peptide the peptide is what the B cell receptor is making the antibody against.
It's part of that
tiled the peptide's like a 15-mer
surface.
Right.
Okay.
So you're pretty restricted in it being an epitope.
Okay.
I always understood a hybridoma as a B cell plus a myeloma cell.
But this isn't, the hybridoma is because people said to me, we don't think you can make hybridomas. We don't think you can do this. And I don't, at this particular moment in time, we don't have $50,000 to do this experiment. That's why I wrote the grant. So I don't want to sack these mice. So what I've done is I've harvested the splenocytes from the mice. I made hybridomas where I fused them to a myeloma cell line. I went to Sloan Dixon. I had to go to Sloan Kettering to learn how to do this because there's nobody here at Columbia who does this. And the woman is amazingly generous. Like, I am so happy that the people who I've asked for antibodies, whether or not they're site-specific or this C3 antibody that is hugely valuable, they've all been really generous in supplying us with this. And then this woman at Sloan Kettering, who runs a core facility to make hybridomas,
Sure.
said to me, "Come on down. I'll give you the protocol. I'll even give you the myeloma cells."
Right.
And then when we took a picture of our first hybridomas 3 weeks ago and sent it to the lady, she gave me even additional comments about like what she thought, whatever, blah, blah, blah. And so now I have hybridomas from our polio immunized mice, our EVD68 immunized mice. And today we're making hybridomas from our Rhino 1A immunized mice.
From single B cells. So monoclonal antibody from single B cells. Okay.
Yeah.
No, the single B cell experiment, which is called LibraSeq, we're not doing because we don't have the money.
Right.
Yeah. So you have the bulk.
Yeah.
I have the bulk splenocytes,
and you could sort them in the future.
Right. That's why I made them hybridomas, is I put them in the freezer to sort them in the future, because I don't want to sack these mice because they have all the answers that I want and just sack them at the ICM facility, right? I want to I have to figure out a way. I had to figure out a way to make it so that I could put the B cells in the freezer, and then when I want them, thaw them out and have them viable. And this is the only way to do it.
Apparently, you can't freeze a spleen.
Is the ultimate result?
No, you cannot freeze a spleen.
Excuse me. Is the ultimate result of your experiments, though, to develop a series of reagents that only distinguish one virus from all of the other viruses?
Well, that's a separate grant, Dixon. That's a separate grant.
For every. Yeah, but if you ever succeed at that, everybody will want your antibodies.
Yeah.
So I need some.
That's one goal.
I need some clarification here. Could you define in sort of, I guess, atomic terms, what a conformational epitope is? And a conformational epitope, what's an antibody binding to?
So that's been a problem, and why we only got a 30 and I didn't get the pot of money the first time I wrote the grant and had to reapply last Monday. So the idea is that you have to fold the whole capsid, and because the loops are intertwined between between the proteins, that the antibody can bind an amino acid on, like, say, loop A of VP3 and loop B of VP2. And if you just had the peptides linear, they wouldn't form the loops and the antibody wouldn't bind.
So that's where my question before was coming from, that if you have a peptide, if you pull out a B cell because it reacts with a peptide,
yes,
does that really tell you what the epitope was in the virus? Or is it that if it's a conformational epitope, your peptide experiment won't work?
No. So it turns out that the conformational epitopes are all a sequential consecutive series of amino acids. So the conformational epitope is much larger than your peptide.
Okay.
So your peptide is embedded within that conformational epitope.
So does that mean that
if you used a peptide to pull out site 1 and you target 15 amino acids within site 1, you will pull out antibodies that neutralize, because that they're within the epitope. So the way Hogel has defined it is that the conformational epitope is like a large square, and your peptide is within that square and it pulls it all out. So therefore, the conformational epitope and linear epitope is the same thing.
So am I understanding correctly, Amy, that this is why it's really important that you're doing the peptides together in a pool, so that
it's kind of
different peptides can come together to make those conformational epitopes. No. And then when you pull it out, you're getting both of them, both barcodes? No? I'm
doing it in a pool because it's easier. I don't know what I'm going to pull out.
So Amy,
the way that Hogel has described it is, and when you look at what is known about polio. So let's say the epitope of the south. So like the epitope of the south wall of the canyon. Okay. It's folded nicely, blah, 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 but it's larger than 15 amino acids.
So will each peptide bind to those antibodies?
They should.
So if you have a conformational epitope made of a couple of different peptides, each individual peptide should still bind to those antibodies.
Yes.
Is that what you're saying?
Yes.
Yeah. So that's.
And so you should actually be able, to capture the same antibody with multiple peptides, which is exactly what people have done. They've done this for HIV, for flu, and for SARS-CoV-2. And so the guy who, there's 4 papers who've used LibreSeq. And the guy is at Vanderbilt, and he has done it. And his first paper, he did it with flu and influenza. He found all of the validated epitopes, and then he found additional epitopes. And then he's recently done it again for SARS-CoV-2. And in his last paper, and which was complemented with a phage display library paper by another group, you could use peptides and you can identify the epitopes and whether or not the spike is in the up or the down conformation.
So they told us, and when we sent this in, you can't get conformational epitopes with LibreSeq. That was it. You can't do it.
Yeah, you can't get conformational epitopes with
why they
peptides.
didn't fund the proposal. And it's not true. And this is an example of how science is so rigid.
Well,
right?
It's not only true. It's not just that it's so rigid. It's that they didn't go back at the literature and look at what was done by Eckard and David Rowland and the FMDB people and Don Wiley for flu and various other things. That's exactly how they did it. Made peptides.
We should have written it in a way that made it clearer.
Wait a minute. Can't you rebut those statements?
No, because it was an RFA, and so the RFA is over.
But I take the blame. We should take the blame because we didn't make it clear enough.
We didn't make it clear enough.
That we said we will be able to get conformational epitopes in Y, but we didn't think we had to do that. I want to also point out that when the paper was submitted, it had really strong opposition. No, antibodies can't cross-react among enteroviruses. No, this is not possible. They even said, go back and read the literature. So Amy did.
EVD68 has a periodicity. It's every other year. And other enteros also have a periodicity.
You mean it emerges in human populations every other year? You should explain that to the general audience, Amy.
Sorry.
You can't use jargon on this show. You have to, come on, people are listening.
Why?
I don't know why, but they are.
Why are they listening?
This is extremely entertaining. This is like writing a grant. I love this show. This is my favorite show of all time right now.
Okay.
We had a brainstorm with you.
Dixon.
No, really. We listen to what went on. If we have to summarize this, we've got 2 retired professors. We've got quiet, Amy. You're not in this conversation right now. We've got 2 retired professors. We've got another one on the way. We've got a young, successful professor, and someone who has written so many grants that I'm beyond taking off my shoes and counting my toes as well as my fingers. And you've brought up all of the problems of writing a grant, submitting a grant, and responding to a grant. And your problems, and you haven't lost faith. That's the other thing. You haven't lost faith.
No, she never gives up. That's amazing. Really, so
here we are trying to come to grips with what your future holds for you should you get funded. I love that. I absolutely love that.
Well, the future, if I don't get funded, I'll be fired.
Well, we know what's going to happen if you don't get funded, so we're not even talking about that. I'll have 2 with mustard, thank you.
Can I just, I want to wrap this up, but I just want to say, Amy, so basically, whether you get antibodies from LibreSeq or hybridomas, then you take them, you do neutralization assays, you find the ones that do cross reactions among entros, and then you identify the epitope. Is that the the timeline? Yeah.
Well, that's not the timeline. That's the plan.
No,
the timeline is open-ended.
Very long timeline.
So to get back to your question, Dixon. So the implication of this.
No, I wanted to wrap this up.
Oh, okay, fine.
It's fine. Well, go ahead,
No,
Amy.
No, this is
No,
just part
it
one.
doesn't
Part
matter.
one. Amy, come back. Come back on the show. We'll help you
No,
write the
it's fine.
conclusions next time.
Maybe I'll have conclusions.
No, I think the
I mean-
identification of these epitopes is going to be really interesting, right? What is this
Absolutely.
monoclonal hitting on two different enteroviruses to neutralize
Absolutely.
them? I think it's fascinating, and Amy's going to bring this forward. Yeah, it's good
Well, it's
stuff.
not just interesting. It's important
That's
for
why it's
understanding
Yeah.
the epidemiology of these viruses, because from what I understand now, you can't do serology and necessarily identify who's been infected with what,
Right.
Exactly.
because there is apparent cross-reactivity.
But-
You can't do a serosurvey looking for D68 antibodies and see reactivity and draw the conclusion that
That's
D68 was actually infecting that person or circulating at that time. That's
right.
not
And
good
that
enough.
has-
That's the whole
And
point,
that
right?
seems super important
Yeah.
for thinking about what you're actually seeing with pathogenesis. If you can't
Yes.
actually say exactly which virus
Right.
someone was infected
Exactly.
with, then how are you actually going to be able
Right.
to make good claims about pathogenesis? This
Well,
And
is
not
Amy-
only
very
that,
important.
how can you actually say which vaccine to produce for next year's go round if you don't even know which one went around this time?
Well, we're not producing like Mike.
But they will. Trust me.
No,
they
yeah,
will.
but I've already had talks about this.
That's
and
very important stuff.
and I would say the people we've had talks. I've spoken to about the future of this and my future and stuff. We all agree you're not going to make we're not going to make another enterovirus specific vaccine like we have a polio vaccine. What a caveat to all of this that we've not addressed at all in this discussion is that I've begun to say that there are discrete antigenic groups of enteroviruses, that polio and EV68 and whatever is in the table, that's one antigenic group. And then another antigenic group might be anchored by Coxsackie B3, and another antigenic group may be anchored by enterovirus D94. And so if you want to make a pan countermeasure, what I am on the way to identifying is the commonality between all of what's in common within that antigenic group. And then you have to have that common denominator for antigenic group A and antigenic group B and antigenic group C, and then you can make a countermeasure. And then it doesn't matter that you really can't do serosurveys because now you have it covered.
So it sounds like to me that you are unearthing a lot of assumptions that people have been making and disrupting the canon in a few places.
That's what science is all about.
Yeah.
And that's what makes it fun.
Exactly.
And that's what makes it a puzzle. And then you piece, like, I come from a line of scientists, so we piece it together, and then it makes you want to come the very next day to do the very next experiment.
That's right.
And
Or even
that's
at night,
why
come back and look at your plates.
I do.
I know you
I do. Sometimes I do.
Listen, Amy, when I was in business, I used to run to work every day. I didn't look at it as work at all. It's not work at all. Somebody in our group of physicians once called research organized play. And I think that's exactly what it is.
Well, at least for me it is.
No, I think if it's not, then you're in the wrong business.
Yeah. I mean, I understand. I'm very lucky. I see the big public health picture. I understand, the goal, and we make a little step forward each day, and but making that little step is what I enjoy. Like, people are like astounded that I still do my own experiments and write grants and write papers. They're like, "What do you mean?" And I was like, "Yeah, I do the experiments, I write the papers, I write the grants, I balance the books."
And you take the credit.
Well, I share the credit, right? I'm sure this was a collaboration with Ian and Niche and Vincent and
No, I understand
Edmund
that,
and Niche and Michaela.
Without you, that wouldn't have happened.
Yeah.
Yeah, as you heard, I didn't believe it.
I've never met anybody in your line of work that created more luck for themselves. You haven't been lucky. You've been absolutely spot on, hardworking. That's my take,
Yeah.
and I'm
Well,
sticking with it.
Thank you, Amy.
yeah. So it's good.
Indeed.
All right. Let's do some picks. I have to leave at 5, so
Okay.
move on to the picks. Dixon, what do you
All right.
have for us?
I'm bringing you up to date with regards to the fusion nuclear generation of unlimited amounts of energy topic that I started last time. This is a new result in just about a month from the group that's at Lawrence Livermore. They use a different way of squeezing hydrogen epitopes together to try to get to the helium and the power release. Remember, they had 1 trillionth of a second's worth of energy released, and they thought that was a big deal. Well, here's a group in England working in CERN's laboratories. They have a tokamak at Oxford, and they have recently announced that they produced 59 megajoules of energy in 5 seconds, which far exceeded the amount of energy within that time frame that the entire tokamak needed in order to operate. So they far exceeded the energy neutral zone and went into the energy production zone. They had to stop their machine because the magnets started to heat up. That's how much energy they produced. So a tokamak is different than the other object that I described before, where the beams of 191 laser beams are all coming together to focus on a small little object about the size of a pinhead. This produces a plasma of deuterium and tritium. And by using electromagnets in a circle that looks more like a donut, it's actually like a torus, you can squeeze this plasma by increasing the amount of magnetism until the plasma reaches a certain density. And at that density, fusion initiates. And that's where they're at right now. And they're very, very excited about this. And they should be, because this is the first time with a stopwatch, you could actually tell how much energy was made rather than a femtosecond or something like that. So I think it's a big deal.
Cool. All right. Amy, what do you have for us?
So I have a kid's book about AFM, and it was written by Rachel Scott and Dr. Laura Grimmer. And Rachel, you and I know well. So her son has AFM and was diagnosed with AFM at 4. And we've been following him from afar to some degree. And it's called Alfredo's Magic Wand. And the wand stops working. And then he goes to find a bunch of friends to help him get his wand to start working again. So,
no spoiler alert here. No spoiler alert.
Yeah.
That's cool.
So, yeah. And Rachel has prolifically written about her child's journey and how it's affected not just him, but how it's affected her entire family. And she's very active in, bringing AFM to the attention of everyone and understanding that, how to prevent it and what we can do.
Cool.
So he had enterovirus 68, right?
Yeah. Yes.
And then he developed paralysis, and now he remains paralyzed to some extent, right?
Yes. He remains paralysis to some extent, but they've had some therapy where he's been able to regain some of his motion and stuff.
And all of this is, she makes very public, so it's not confidential medical information.
No, she's documented, she actually had a child right after he was diagnosed with AFM. She documents how, he was in the hospital for basically a year, and him coming home, and all of the therapy, the nurses, the whole way that they've rearranged their lives. him, like all of the therapies that they've given him and have led to him regaining some motion, including some surgeries. And, you know, like when he was off the ventilator and when he can chew and swallow and various other things. Yeah.
I follow
and him
her
in the swimming pool and everything. Yeah.
I follow her on Instagram. She talks all about that. Yeah. Just pictures and so forth.
Yeah. I mean, it's, yes.
I mean, she'll show a picture of him and she'll say, oh, tracheotomy tube is out today. You know, I mean, she wants to share it. Yeah.
Right. And then she tells you all about the when he slips back, like, you know, he's not really able to clear some respiratory infections very efficiently. And then he has to go and he'll go back to the hospital. And then she's talked about like they've had slumber parties where all of her children live in one room all together, and the night nurse coming or the night nurse not coming to, you know, it's the first time that he would be able to do these kind of things, just regular childhood things.
Cool. I mean, the book is cool.
The book is really great. Like, I have my own copy and everything. Good. So, yeah, everybody should have a copy.
Brianne, what do you have for us?
I have an article called What Happened After the Chickenpox Vaccine. And this is sort of a story about the details of some development of the chickenpox vaccine and approval and sort of some of the mandates for chickenpox vaccination and sort of how that all happened, with some comparisons to vaccinating kids against COVID-19. And it's a really fascinating story, especially because I'm in sort of the age bracket of people that they talk about who, you know, were sort of in that last group of people who had the chickenpox and sort of had the chickenpox, and then suddenly there was a vaccine, and it was sort of, the whole thing seemed a little shocking. And I think that the last sentence of this article really brought a lot of it home for me, which was, perhaps it's only natural to long a little for the before time of chickenpox, when the question was whether we would be able to wipe out a disease, and not, once we had the capability, whether we would choose to do it.
Indeed. It's too bad.
And so the whole way that the chickenpox vaccine came about and the mandate for kids is all detailed here.
Cool. That's great. Rich, what do you have for us?
Well, actually, Brianne's picked more or less segues into this. And for those of you who were looking for some SARS-CoV-2 on TWiV today, here it is. My friend Nassim Masachi sent me a YouTube video of a grand rounds at the Mayo Clinic by Gregory Poland, who's an immunovirologist of some repute. This was January 20th. So it's pretty up to date. It's in the Omicron era, and it's his sort of current perspective on, as of that date, where the pandemic, what the status is. You'll see it's timed at an hour and 40 minutes, but I think the lecture was an hour or less, and there's a lot of questions afterwards. I didn't actually follow all the questions, but it's a nice summary. He at times makes some pretty sweeping generalizations that I think are a little more nuanced than that, but that's the nature of the summary. One of the real take-home messages is that he's very clearly frustrated by the fact that our society and our culture has the capability, that this didn't have to happen, okay? That we have the capability to, even once it started to happen, bring it under control, and we're still messing up with vaccination, appropriate other precautionary measures, okay? At any rate, I found it a nice summary where we stand.
Great.
He's a very forceful speaker.
Thanks, Nissin.
Yeah.
My pick is an article by Laurie Garrett in FP. What does that stand for? Foreign Policy, sorry. not a journal I look at often, but I somehow I got on her email list, and whenever she writes, she sends it out to us. And this one is very interesting. It's called Economists Are Fueling the War Against Public Health. So you may have heard about this 62-page study that's been released, getting a lot of attention. It claims that all the regulations, masking, lockdowns, school closures, had a minimal effect on SARS-CoV-2 infections, deaths particularly. And she goes through it and basically says, well, it's an example of motivated reasoning, indulging in scientific cherry picking to prove a preferred thesis about public health. And it is, she goes through it. I'm not going to repeat it for you. You should just read it. But it's essentially garbage and not worth reading, but you should read her article about it. It's just put together by people who have a political agenda, and it's being bandied about by them. Hey, we don't have to do all this stuff. Look, this proves it. Well, it doesn't prove anything. It's just nonsense. They didn't actually do anything. They just went through the literature and pulled out some numbers. And she said they pulled out the numbers they wanted to. I mean, for example, one of the supporters of this supported the Great Barrington Declaration. Remember that?
I was just thinking about that.
Yeah. So that's really good. She did a great job with that. You should check it out. All right, that's TWiV 866. You can find the show notes at microbe.tv/TWiV. Send us your question, comments, picks of the week, TWiV@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Your contributions are federal U.S. tax deductible. Amy Rosenfeld is at Columbia University. Thanks for joining us today.
My pleasure.
And good luck with that cool stuff, which I think is amazing. And this story really illustrates many aspects of science, as we have said. Dixon Despommier is at trichinella.org, thelivingliver.com.org.
Howdy.
Thank you, Dixon.
This was an amazing skull session. I loved it. Amy, I hope you weren't offended by anything I said. I tried not to.
Of course not. I love you, Dixon. I know.
No, it's the same.
We know it comes from the heart. I'm not.
Just keep on. And by the way, I appreciate Vincent's sentiments, but I don't think you need luck. You're a hard worker. You don't need luck, and that's a good thing about what you do.
We should say that when Dixon was outside my office, he had a little desk there. Amy, whenever she came to talk, she would always stop and chat with Dixon, right?
Always.
Yeah.
Always.
Often
I learned so
for a
much.
while.
No, I learned a lot. I miss those sessions.
He tells great stories.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Thanks, Amy. This was fun.
Oh, yeah, it's good.
And Brienne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brienne.
Thanks. It was great to be here. Thanks, Amy. I'm going to ask you some things about some of those papers so I can read more about this a little bit later.
I mean, it's definitely revolutionized our thinking.
Oh, yeah. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.